So good evening to you all. The last time we met, we had finished with the metabolism of lipids. We started with metabolism of carbohydrates. We did metabolism of proteins. And then we went on to do metabolism of lipids. And today we are coming to do metabolism of nucleic acids, specifically metabolism of nucleotides. And this will be the last lesson for the semester before you prepare to take your exams. Today, I had put some questions online for you on the first part of the program, which was the study on enzymes, enzymology. So if you go to the LMS, you would find that there are some questions there on enzymes for you to try your hands on. Over the Easter holiday period, from this week onwards, I'll be putting questions under each of the sections so that you can try your hands as you prepare for your exams when you come back after the Easter holidays. With that said, let's dive into the metabolism of nucleotides. So you had studied the basic units of nucleic acids to be nucleotides. And these nucleotides have nucleobases or nitrogenous bases. And these nitrogenous bases are divided into two major groups, the purines and the pyrimidines. So we are going to take a look at the metabolism of purines. And after that, we will look at the metabolism of pyrimidines, which is similar to what will be happening to the purines. So what we are looking at currently is the sources of the various atoms that are present in the purine rings. So we see, for instance, N1, this nitrogen, and this whole portion, the double bond, the single bond, all of this side comes from aspartic acid or aspartate. That's an amino acid. Similarly, N3, N9 also come from an amino acid, glutamine. Then we have C4, C5, N7 coming from the amino acid glycine. We have seen all these compounds before. Then we see C2, C8 coming from tetrahydrofolate, which we saw as one of the coenzymes when we were looking at some coenzymes. We commonly refer to it as folic acid. Then, of course, there is carbon-6, which is coming from carbon dioxide. So you see these sources, the amino acids, the folic acids, carbon dioxide, providing all these components to come together to form the purine ring. Now, so that process in detail, as we are looking at it now, so that process in detail, you see that the final product, inosine monophosphate, is the parent molecule, or let's say the precursor for the formation of the purine ring, or for the purine nucleotide. This is inosine monophosphate, which is the final product. Now, you see that, first of all, the nucleotide structure consists of a ribose sugar, a phosphate group, and then a nitrogenous base, which will be on carbon 1. And you see this reaction path is starting from ribose 5-phosphate. And we had seen ribose 5-phosphate being produced when we were studying the metabolism of carbohydrates. We saw the pentose phosphate pathway, which is also referred to as the hexose monophosphate shunt, from which we saw the production of ribose 5-phosphate. So we can now relate the connection between carbohydrate metabolism and metabolism of nucleotides. Right from the beginning, we see that that ribose 5-phosphate comes into the synthesis of nucleotides. So we see that, first of all, this ribose 5-phosphate, which was produced in the hexose monophosphate shunt or the pentose phosphate pathway, first of all, gets phosphorylated. So you see ATP coming in this reaction. And so there are 2 phosphate groups that get attached on carbon number 1. Now, remember, all along, we have been seeing these similar reactions where in the beginning of the reaction path, we see the phosphorylation of a compound. And I mentioned that this type of reaction, this phosphorylation, you will see often. And this is a form of activation of a molecule or a form of activation of a compound to go down a reaction path. We are seeing it here again. ATP is releasing 2 phosphate groups, a diphosphate or pyrophosphate, which, when conjugated to ribose 5-phosphate, activates this molecule to go down the reaction path. Now, then we see the sources of the atoms that are in the purine ring: glutamine, glycine, the tetrahydrofolate, aspartic acid, now coming into action. First, you will see that glutamine, which is an amino acid, donates its amine group on carbon 1. So see this as deamination. So here again, you see that there is that connection of the metabolism of amino acids with the metabolism of nucleic acids. So you see glutamine, an amino acid, gets deaminated, and the amine group, which is removed from glutamine, is transferred onto the carbon 1 of the ribose 5-phosphate. So you can see it as a transamination, but this time around, the transfer of the amine group is coming from glutamine onto the ribose 5-phosphate. Then you see glycine now comes in. Glycine now comes and gets conjugated onto the amine group, which just got conjugated to the ribose 5-phosphate. And the reaction continues. Tetrahydrofolate, commonly we refer to it as folic acid, also comes in and comes to add some ketone group or C double bond OH group onto the glycine residue, which just got attached. And the reaction, this reaction path continues all the way. So now you have seen the 3 amino acids in action. Glutamine coming in first to give the amine group. Glycine comes in, gets conjugated on that amine group. Almost like that is like a peptide bond which is being formed on the ribose 5-phosphate. And then tetrahydrofolate comes in to continue forming the ring. But that is just the first part of the ring. That is the six-sided ring. And then it continues to form the five-sided ring. And by the time this whole reaction comes to the end, you'll see that you are now having inosine monophosphate. So the ribose 5-phosphate. You see that from here, the first step, we have shown the ribose 5-phosphate. Here, first step, second step, third step, fourth step. As it got complicated, we just write ribose 5-phosphate in place of this whole molecule to make it simpler. So we just put ribose 5-phosphate to show just the formation of the ring onwards. So onwards, you are not seeing the sugar molecule, but you are only seeing the sugar molecule being written in words, ribose 5-phosphate all along. Then you also see that along the line, you see there is a release of fumarate. Fumarate too, we have seen as an intermediate in the tricarboxylic acid cycle, the TCA cycle. So here again, you see a connection with carbohydrate metabolism, because this fumarate can then go into the TCA cycle. So for you to appreciate how these amino acids together with folic acid will come together to form the nucleotide, ribonucleotide. Now, you go on further. So now you have your inosine monophosphate. This inosine monophosphate goes into the formation of two nucleotides, which is adenosine monophosphate and guanosine monophosphate. Remember, the purines are two. We are looking currently at the synthesis of purines, and these purines are two. One is adenine, the other is guanine. So if we have the final product, which we saw from the previous slide, we saw it to be inosine monophosphate. So this is a continuation of what will happen to inosine monophosphate. So inosine monophosphate, again, the reaction continues. Again, you see aspartic acid, which is transferring its amine group onto inosine monophosphate. So what you see is that, so this is aspartic acid, and so this is the carboxylic end of that amino acid, and you have the amine group of that amino acid getting attached on inosine monophosphate in place of this C double bond O. You see the aspartic acid coming to get conjugated to that molecule. And you see this is the side chain, and the side chain is having a second carboxylic group in it. So then you see that from here, you see the amine group is cleaved. So you see the amine group is cleaved onto the inosine monophosphate now to form adenosine monophosphate. And again, you see fumarate, which is an intermediate in the TCA cycle being released. And so this goes into the TCA cycle. Now, for guanosine monophosphate, you see that first of all, there is some form of hydroxylation. This can say this is an oxidation reduction reaction occurring at the same time. So you see that oxygen from water gets attached on C2, carbon number 2. And then the N3 also gets reduced. So you see from here, there is no hydrogen here, but here you see a hydrogen. And this hydrogen is coming from this coenzyme, the nicotinamide nucleotide, which is involved in the transfer of protons in that oxidation reduction reaction. So this N3 is also reduced. And then further, glutamine comes in to transfer its amine group onto this carbon number 2. So this carbon number 2, which first got oxidized. So it's a form of activating that carbon to now go into a conjugation reaction with glutamine. So the glutamine now transfers its amine group onto the molecule to give you guanosine monophosphate. Now, you have adenosine monophosphate, you have guanosine monophosphate. So that means you now have the purine and the purines, let's say purine nucleotides, both of them, adenosine monophosphate, guanosine monophosphate, which you find in the nucleic acids. All right, now going forward, the formation of diphosphates and triphosphates. So this is simple. We have seen the formation of these monophosphates. So that nucleoside monophosphate only undergoes two-step reactions to form a triphosphate. So for instance, so what it means is that if ATP donates one phosphate group onto these nucleoside monophosphates, then you'll be having a nucleoside diphosphate. And the second step, ATP again donates another phosphate group onto this diphosphate. You now have a triphosphate. And so you'll be having adenosine triphosphate and guanosine triphosphate. So this is simple. And take note that the enzymes which are catalyzing these reactions are, the first is what? Nucleoside monophosphate kinase. We have seen these over and over. And I mentioned earlier in our lectures that if you see any enzyme with a name kinase, it means it is responsible for the transfer of a phosphate group from one molecule onto the other. You see the same thing here. Nucleoside diphosphate kinase, responsible for the transfer of a phosphate group onto the diphosphate, the nucleoside diphosphate, to give you the nucleoside triphosphate. Now, so a bit of practical application of this knowledge of the synthesis of purines. So there are these class of antibiotics referred to as sulfonamides, sometimes being referred to as sulfanylamide. So sulfanylamide is a drug. And if you look at the structure of sulfanylamide compared to para-aminobenzoic acid, there is a structural similarity. Now, the difference you see is that you see a sulfoamide group, some sulfate group with an amide group, which is replacing the carboxylic group in para-aminobenzoic acid. Now, you see in bacteria, this reaction path for the formation of folic acid, tetrahydrofolic acid. Remember, we saw that tetrahydrofolic acid is responsible for cell growth, cell reproduction, cell proliferation. You remember I mentioned that folic acid is needed in the developmental stages of the fetus. And that is the main reason why usually this folic acid is being prescribed for pregnant women, because the deficiency of it can cause neural tube defects, that is, defect in the growth or the formation of the brain. And so you see this similar synthesis of folic acid in bacteria being important in the activities of the bacterial cell. And the beginning of the synthesis of folic acid in bacteria starts from the para-aminobenzoic acid reacting with a compound dihydroxyl diphosphate, which further goes down the reaction to give you the tetrahydrofolic acid. Now, sulfonamides, looking at the structure, the sulfonamides is a structural analog. So it looks all just like the para-aminobenzoic acid, and so acts just as if it is a para-aminobenzoic acid. And when introduced in the body, it goes to inhibit this first enzyme, dihydroxyl synthase, because it's acting as if it is a substrate to the enzyme, as if it is para-aminobenzoic acid, but it is not. And in its place, in place of the para-aminobenzoic acid, if the sulfonamide comes in, it will be acting as an inhibitor. The reaction will not proceed to the final stage of the synthesis of tetrahydrofolic acid. And that is the principle by which these drugs work as antibiotics to inhibit the synthesis of folic acid by microorganisms. Now, as some other structural analog can be methotrexate, which is a common drug used in the treatment of cancers. And this drug also will be having effect on the synthesis of folic acid. So we're acting on, say, dihydrofolate reductase for this reaction to continue. So you see that that reaction will not proceed. And what it means is that cancer cells will not be able to reproduce and will not be able to multiply to be forming tumors. And with time, you would have been able to destroy these cancer cells. But of course, there are side effects. So if you are inhibiting the synthesis of folic acid, which we say is responsible for cell growth, the cell cycle to proceed normally, then it means that if you are inhibiting this process, then it affects, say, the development of the red blood cells, the erythrocytes. So that process of erythropoiesis is going to be inhibited, and that will be causing some form of anemia. Same way, if it's inhibiting the synthesis of follicles, the production of follicles, which is responsible for the hair that grows, of course, then it will be causing some baldness. It will cause some inhibition in the growth of the epithelial cells. And so then you have some kind of scaly skin also being a symptom of such chemotherapy. So that is just a practical application of this knowledge of the synthesis of nucleotides or nucleic acids as a whole. Now, what we saw earlier about the synthesis of these nucleotides is what is referred to as de novo synthesis, which means synthesis of these nucleotides from smaller molecules from scratch. So you see that the various components were coming from different amino acids, in addition to carbon dioxide and then the folic acid. But there is also a simpler pathway of the synthesis of purines. In fact, we'll also see that that salvage pathway also exists in the synthesis of pyrimidines as well. So in general, in the synthesis of these nucleotides or the nucleobases, which is the salvage pathway. And this simpler pathway is just about the conjugation of an already existing nitrogenous base with an already existing ribose 5-phosphate. So what does that mean? Now, this is like recycling. So if you have some nucleic acid, which is broken down, so you know the nucleotide is made up of the phosphate sugar and then the nitrogenous base on it. So if there is some breakdown of this nucleotide such that the phosphate group is hydrolyzed away, the nitrogenous base is hydrolyzed away, which means you are having the three components standing on their own. You have the phosphate group separated, you have the sugar separated, and you have the nucleobase also separated. Now, if you have that base getting conjugated on a ribose 5-phosphate, which is produced from the pentose phosphate pathway, that is simple. And that is a form of recycling of these molecules, and it is referred to as the salvage pathway. So you see here first, you see adenine, which is getting conjugated onto the ribose 5-phosphate to give you adenosine monophosphate. And you see here the enzyme which is acting is adenine phosphoribosyl transferase, because there is some transfer of a phosphate group. So a pyrophosphate is given out from ATP, and then the single phosphate group comes on a ribose to give you a ribose 5-phosphate. A similar reaction is occurring with guanine. Guanine gets conjugated on ribose 5-phosphate, and you have the guanosine monophosphate. And you also have an intermediate in the synthesis of these purines, hypoxanthine, which can also undergo this reaction to now give you the inosine monophosphate. So if you have inosine monophosphate, which we have seen earlier as the parent molecule for the formation of these purine nucleotides, then of course from this inosine monophosphate, you can have the synthesis of adenosine monophosphate and guanosine monophosphates. Now, the salvage pathway is usually found in the red blood cells and brain cells where you do not have the de novo synthesis being operative. So, of course, you know that erythrocytes do not have nucleus in them, but that is the adult erythrocyte. The adult red blood cell does not have nucleus in it because, of course, there's a need to have a bigger space for hemoglobin so that that cell will be able to carry enough oxygen from the lungs to the tissues and then carry carbon dioxide back to be exhaled from the body. But in the life cycle of that erythrocyte, from the beginning of the production of the red blood cell, that process of erythropoiesis, in the beginning, the young red blood cells have nucleus in them. But along the line, that nucleus is lost before you have the adult red blood cell. So in that process, the nucleus which is present is having this the nucleic material being synthesized through the salvage pathway. And it's the same thing which happens in some brain cells. That's what happens in brain cells. So in the nervous system, you have the synthesis of these purines, these nucleic acids coming from the salvage pathway. Now, we have talked about the synthesis of these purines. And now briefly, we will talk about how this process of the synthesis of purines is regulated. How will that process be stopped when it is supposed to stop, and then when it is supposed to continue? How is that regulation done? Now, many of these reaction pathways are regulated by what is referred to as the feedback mechanism. It's a negative response which will cause the reaction path to stop. So what it means is this. So if you look critically in these reaction paths, you see the enzymes that are responsible for each reaction path are shaded in green. So the phosphoribosylpyrophosphate synthase, first one, the phosphoribopyrophosphate glutamyl aminotransferase, on and on and on. You see all of them: synthase, carboxylase, synthetase, all of them over here. Adenosuccinate lyase, formyltransferase, cyclohydrolase. All these enzymes are shaded in green. Now, you see that adenosine monophosphate and guanosine monophosphate are products that are realized in this process. So what I mean is that in the first reaction, you see that adenosine monophosphate transfers two phosphate group, a pyrophosphate, onto the sugar, which means that you will be left with adenosine monophosphate. So adenosine monophosphate is a product because two phosphate groups comes from ATP onto the sugar to activate it. And so you have adenosine monophosphate. Now, this adenosine monophosphate, which is a product. So in some cases, you can also have guanosine triphosphates, which can be acting. But these products, the adenosine monophosphate and guanosine monophosphate, now become inhibitors to the second enzyme, phosphoribosylpyrophosphate glutamyl aminotransferase, which is responsible for the transfer of the amine group from glutamine onto the ribose 5-phosphate. The first transfer of an amine group before glycine comes onto it. You see the enzyme which is acting there. You see adenosine monophosphate and guanosine monophosphate acting as inhibitors. That is a form of a feedback mechanism. So what it means is that if you are having a lot of these ribose 5-phosphates becoming diphosphates, which means there will be more of glutamine, the transferring amine group for the reaction path to continue. But once you are having more of this adenosine monophosphate and guanosine monophosphate in the system, then it goes to inhibit the enzyme to say, hey, we are getting too many in the system. So stop, relax. And so you see that the whole process of the synthesis of inosine monophosphate, which will go into the synthesis of the purine nucleotides, will be inhibited. Now, so that is about the negative aspect of it. How about the activation of the process itself? So for instance, in order for this reaction to occur, logically, you should have enough of the phosphoribosylpyrophosphate. Of course, that is what will go into the very first reaction. The phosphoribosylpyrophosphate synthesis is the enzyme that's responsible for the first reaction. And so you need that phosphoribosyl pyrophosphate, first of all. The phosphoribosyl pyrophosphate, which is this. So phosphoribosyl pyrophosphate, this, you need a lot of this in this. You need enough of this in the system for glutamine to be transferring amine group onto them, and then glycine comes, and then the reaction continues on and on and on. If you don't have enough of this phosphoribosyl pyrophosphate, then of course the synthesis is not going to be at a higher rate. If you have less, you have lesser rate of the synthesis. If you have more, you have a higher rate of the synthesis. Similarly, the availability of the ribose 5-phosphate. So this is the ribose 5-phosphate itself. So which means that the synthesis of the ribose 5-phosphate from the pentose phosphate pathway, what it means is that process of the ribose 5-phosphate, if there is a defect there, it is going to affect the synthesis of the nucleic acids, or the synthesis of the nucleotides, which will go into the synthesis of the nucleic acids. Again, I've talked about this, the availability of the enzyme. This enzyme, phosphoribosyl pyrophosphate synthesis. There must be enough of this enzyme. So this will then go to protein synthesis. So you remember your protein synthesis, the process of transcription, translation. In that process, there too, the normal process means that there is going to be enough of the enzyme available to catalyze the reaction. But if there is a defect in the synthesis of this protein, the protein which becomes the enzyme, the phosphoribosyl pyrophosphate synthesis, then of course, this reaction is going to be inhibited. So you see the interconnection of all these reactions happening in the body. All these things are happening at the same time. And so the synthesis of proteins, the synthesis of carbohydrates, that is the ribose 5-phosphate, all of these will be having effects one way or the other. If there is a defect somewhere, it goes to affect the other. All right. Now, regulation. Okay, so again, the continuation of the regulation. I've talked about the feedback mechanism. So the feedback here, we are seeing that the adenosine monophosphate, which is the final product, adenosine monophosphate and guanosine monophosphate, the final products. So as we saw, so from here, we saw right from here that adenosine monophosphate, guanosine monophosphate will be inhibiting this enzyme. And we see that inosine monophosphate right from here, the inosine monophosphate, when the continuation of the reaction to the last end, you see the final product being adenosine monophosphate and guanosine monophosphate. These are the final products. And these final products, for instance, the adenosine monophosphate will be acting as an inhibitor to this first enzyme, adenyl succinate synthetase. And so once it goes to inhibit this, so it's telling the enzyme, hey, we are enough in the system. We are enough. And so it goes to inhibit this enzyme so that the conversion of inosine monophosphate down the path to give you the adenosine monophosphate will be reduced. Similarly, guanosine monophosphate goes to inhibit this first enzyme, inosine monophosphate dehydrogenase. So you see, it is either you have this final product, adenosine monophosphate and guanine monophosphate, inhibiting these first enzymes that you see in the conversion of inosine monophosphate, or back here, you see them inhibiting the enzyme in the second reaction, in the reaction path to the formation of inosine monophosphate, which means here, when they are inhibiting inosine monophosphate, the production of inosine monophosphate itself is going to be reduced. If that does not fully succeed and inosine monophosphate is already produced, still, they can still come and now inhibit the enzymes that are responsible for the conversion of inosine monophosphate down the path to form them, the product themselves, which are now the inhibitors. Now that's about the regulation of the synthesis of these purine nucleotides. Now, so what we have seen so far has been the synthesis of ribonucleotides. Because if you look at the sugar right from the beginning, you see this is a ribose. You see it's a ribose 5-phosphate. The ribose 5-phosphate is what you find in RNA. But you also need to have the synthesis of deoxyribonucleotides. So these deoxyribonucleotides are what you find in DNA. We have produced so far the biosynthesis which we looked at. We saw the ribonucleotides which will be in RNA. But the conversion of this is just a reduction reaction on. So you have a reduction of carbon 2, which is carrying the hydroxyl group. So this will be reduced, this hydroxyl group will be reduced to just a hydrogen in the deoxyribonucleotides, which will be in DNA. And in that reaction, of course, it is a reduction reaction. And so you have a ribonucleotide reductase. If you are classifying this, of course, this will be an oxidoreductase. The first group of the enzymes which we saw. And even if you look further, you see that this enzyme is having a coenzyme, which is thioredoxin. Thioredoxin in its reduced form in this reaction now loses, so it carries that hydrogen which will get attached onto the carbon number 2 for that reduction reaction to occur. And whilst it does that, itself gets oxidized, and so it becomes thioredoxin in its oxidized form. Here you see the hydrogen that it is carrying. And when it gives out that hydrogen, that proton, then it becomes oxidized. And even that conversion itself is also being carried out by another enzyme, which is thioredoxin reductase, which itself too has the nicotinamide dinucleotide phosphates as a coenzyme. So this enzyme is having the NADPH as a coenzyme. Of course, again, a practical example of the application of this knowledge, where you have hydroxyurea, which is used in the treatment of cancers because it inhibits the ribonucleotide reductase, this enzyme, the ribonucleotide reductase. So if you have hydroxyurea acting as an inhibitor to ribonucleotide reductase, it means that it goes to inhibit the synthesis of deoxyribonucleotides. And if it inhibits deoxyribonucleotides, it means it is inhibiting the synthesis of DNA. And once the DNA of these cancer cells are being inhibited, then of course, their growth, their proliferation, their reproduction is going to be inhibited, so that there is that inhibition of the formation of tumors. So that is another example of the application of this knowledge. Now, so far we have looked at the synthesis, that is the production of these purine nucleotides. But let's briefly look at the degradation, the breakdown. So earlier we have been looking at anabolism. Now we look at catabolism, the breakdown of these nucleotides. So you see, this is adenosine monophosphate, this is guanosine monophosphate, and we are going to look at the different parts. So if you take adenosine monophosphate, for instance, either the first thing which happens on adenosine monophosphate is what? Deamination, so that you have the amine group removed from that adenine base. And so then you'll be left with the inosine monophosphate, which is the parent molecule, which then goes down to now give you inosine, which means that from inosine monophosphate, the phosphate group is removed. So first, amine group is removed, deamination, and then second, you have the removal of the phosphate group to now give you just a ribose, which is conjugated to that inosine to now give you just you now have inosine, the whole molecule being inosine, because you have, first of all, on these carbon 6, you have the amine group removed. So now you have replaced with oxygen, which is coming from water. And then you are left with, and then a phosphate group is also removed from the ribose, and then you are left with just the inosine. Or the first thing, so you see that here deamination occurs before dephosphorylation is occurring, but here is the opposite. So you either have this or this. So what is happening here is that, first of all, you have dephosphorylation happening, and that dephosphorylation just gives you adenosine. Just the phosphate group is removed, the amine group is still there before deamination occurs. And then still all of it is ending up with inosine. And inosine now comes down the reaction. You see, we saw hypoxanthine as one of the intermediates in the reaction part earlier. And this hypoxanthine now comes down. See, the final product in all of this is uric acid. So uric acid is a final product in this degradation. For the guanosine monophosphate, you see that, first of all, there is that dephosphorylation. So dephosphorylation occurs, but after the dephosphorylation, you don't have directly deamination to give you inosine, but rather the reaction continues such that there is now the cleavage away of the ribose. So the ribose is cleaved away, and so you are now left with just the guanine. And this guanine now gets deaminated to now give you xanthine. And then this xanthine now goes down into the synthesis of uric acid. And so xanthine is referred to as the merging point for the degradation or the catabolism of the purines, which is the adenosine monophosphate, guanosine monophosphate. So guanine goes through this part, adenosine goes through this part, but all of them will come and meet at xanthine. That is the merging point. Then from xanthine, there is that oxidation which occurs, and you have the product being uric acid, the final product, uric acid. But that is in mammals like us, you find the final product being uric acid. But in other organisms, so for instance, in some fishes, you see this uric acid going further down the reaction to now form the further breakdown to give you what is referred to as the allantoic acid. In some amphibians and mollusks, so these some snails, some oysters, yes, you have the breakdown further coming to form urea. And in some marine invertebrates, it will even go further to form ammonia. So that is what occurs. Apart from what happens in us mammals, you will see that in other organisms, uric acid further goes down the path to get broken down into smaller components or get converted into other compounds before they are excreted from the bodies of these organisms. Now, what is the health implication of this? For instance, so you see the elevation in uric acid. So uric acid, which is the final product from that degradation. If you have elevation of such uric acid in serum, which of course will get excreted also in urine. You have a condition which is referred to as hyperuricemia, and the accumulation of the salts. So the salts of uric acid. So for instance, sodium urate, which is, you can say that sodium urate, that is a salt, the sodium salt of uric acid in the joints will be causing some arthritis. So what happens is that these sodium salts go to soften the cartilages that are in between the bones of the joints. And as these cartilages are soft, then you have these bones touching each other, having more friction. And so you now have a problem with adequate lubrication, and that gives pain at the joints. You can also, this can also be causing, the deposits of these salts can also be causing some kidney stones, even with the liver. If so you can either be having hepatic stones or renal stones. So you have the accumulation of these things causing the blockade of ducts in these organs. Now, what can cause such a thing? So you see, we have looked at feedback mechanism. So the lack of such feedback mechanism regulation of the phosphoribosylpyrophosphate pyrophosphate synthesis from ADP and GDP. So adenosine diphosphate, guanosine diphosphate, that lack of feedback mechanism. Also from adenosine monophosphate and then guanosine monophosphate on glutamyl amidinotransferase means that there will be that more formation of the purine nucleotides, and their breakdown further to uric acid will be increased in the system. So that lack of regulation can cause such a thing to happen. Now, there is this compound, there is this drug, allopurinol, which is an inhibitor of the enzyme xanthine oxidase, which we have seen earlier. We are seeing it here again, which is responsible for the conversion of apoxanthine and xanthine to uric acid. And as it inhibits this enzyme, then of course it reduces the amount of uric acid which is produced in the system, and thus it will reduce the formation of the salts of uric acid, which can get deposited in the joints to be forming the condition gout, or even the elevation of the compound uric acid in serum and in urine, which will be causing hyperuricemia. So allopurinol can be used to manage such a condition as an inhibitor to xanthine oxidase. Now, there is also this health implication referred to as the Lesch-Nyhan syndrome. The Lesch-Nyhan syndrome is an inherited disorder. It's a recessive disorder. It's on the X chromosome, and so this comes from the mother. Now, you see this condition in males. In females, that you have this recessive gene, the females are just going to be carriers, and so you don't have any symptoms for females. But this is a serious disorder in males. It's a rare kind of a disorder, and the cause of this is that you have, so we have seen the two forms of synthesis of these purine nucleotide. We have seen the de novo synthesis. So vertically, we are seeing the de novo synthesis all the way to inosine monophosphate, and then the breakdown all the way to uric acid. So de novo synthesis, first of all, giving you the inosine monophosphate, from which you now have the synthesis of guanosine monophosphate and adenosine monophosphate as de novo synthesis. And then the breakdown, which the degradation to uric acid is showing here. Then the salvage pathway. because the salvage pathway, we saw that it's just a recycling pathway where you already have some guanine in the system, guanine, which will just get converted to guanosine, because you just have an attachment to a sugar, and then a phosphate group is attached, and so you have that. Now you have these, you have the enzyme hypoxanthine phosphoribosyl transferase, and this enzyme is responsible for these recycling. So you see, guanine can go into the formation of guanosine monophosphate. Hypoxanthine can go back into this formation of inosine monophosphate. Now, if you have a deficiency of this enzyme in the system, what it means is that a lot of the hypoxanthine, if this cannot go back in that recycling part, it means more of it will go into the production of uric acid. And so that will be causing an increase in the amount of uric acid in serum, as well as the production of its salts, which can get deposited in the joints. A similar thing will be happening also with guanine. If it cannot get recycled to give you guanosine monophosphate, which can go further to the production of inosine monophosphate, then you see a lot of it will be going down in the path of the breakdown into uric acid. So you have excessive production of uric acid, and this will be happening in juvenile. So this sometimes is referred to as a juvenile gout, because it's an inherited disorder which will be occurring in a child. And that overproduction of uric acid can have serious effects on the nervous system. So you have this nervous system impairment. And you will see, so what it means is that it can be causing some form of mental retardation as well. And once it is having such an impairment of nervous system, it can also be having effect on the coordination of the limbs, the hands, the legs. It has effect on, the nerves working together with how the hand should move. And so there is that kind of a dyslexia. The person is not able to move his hands properly if he wants to hold something, or lift the thing, kind of. And you see also a self-injuring behavior in such individuals. So you see them biting their fingers excessively, and then even their lips, that is bleeding, and they are still biting. You will see such a rare condition occurring in some individuals. It is referred to as the Leishman syndrome. Now, pyrimidine. So what we have seen so far with purines is similar. It's a similar thing which is going to be happening with pyrimidines as well, except that for pyrimidines, you are seeing a smaller number of compounds which are serving as the source of the parts of these purine rings. So you see here, you see only glutamine and aspartic acid as the amino acids, but for purines, we saw glutamine, we saw aspartic acid, and we also saw glycine as 3 amino acids. Then apart from that, apart from the carbon dioxide, which is giving carbon in that ring, we also saw the tetrahydrofolate also giving some parts to come into the formation of the ring. Here, you are having a lesser number of molecules donating these parts. Now, so that's process of biosynthesis. Now, the difference between the major difference between the biosynthesis of purines and pyrimidines is that if you go back, so let's go back to the synthesis of purines, and you see something. You see that the synthesis of purines is taking place on the ribose sugar. So the ribose sugar is first of all, this ribose sugar gets phosphorylated, And then glutamine first, transfers its amine group, and then you see that the formation of the ring is happening on the sugar. So you see continuously ribose 5-phosphate is there, ribose 5-phosphate, all the way ribose 5-phosphate. So the ring is being formed on the sugar for the synthesis of purines. But now let's go to the synthesis of pyrimidines, and you see what is happening. You see that there is no sugar. Right from the beginning, the synthesis is not happening on sugar at all. You only see what? Carbon dioxide, glutamine coming together to give you carbamoyl phosphate. And that carbamoyl phosphate, aspartic acid, the next amino acid, gets conjugated on that carbamoyl. So the phosphate group is replaced by the aspartic acid. And that is also looking like more like a peptide bond, which is formed between the glutamine residue and the aspartic acid residue. And the ring, and this is only one 6-sided ring for pyrimidine. So this is much easier to form. And the reaction goes down the path forming orotic acid, which is represented here as orotate. That is orotic acid being written as orotate there. Now, so you see that major difference. Purines are synthesized on the ribose sugar, but pyrimidines are synthesized separately. So after you have this orotic acid being synthesized, before you now have the attachment of what? Ribose 5-phosphate. And then that reaction continues to give you uridine monophosphate. And uridine monophosphate then goes further to give you the deoxy thymidine monophosphate, and then cytosine triphosphate as the end products that we are seeing here. So you have seen that major difference between the purine synthesis and the pyrimidine synthesis. Then we also see that yes, right from the beginning, we can see that there is either some activation or inhibition, so kind of a regulation also can happen. So the carbamoyl phosphate synthase to this enzyme, this first enzyme, is activated by the phosphoribosylpyrophosphate. So phosphoribosylpyrophosphate, what it means is that the sugar with a double phosphate group on it in the system will be activating this enzyme, activate this enzyme in order for the start of the formation of the pyrimidines. And similarly, you have the end product, the uridine diphosphate, uridine triphosphate. So you have uridine diphosphate and uridine triphosphate, which will be acting as inhibitors. And that is feedback inhibition. If you have increased amounts of uridine diphosphate and uridine triphosphate as products along the reaction path, these now act as inhibitors to this enzyme, and that is feedback mechanism. And similarly, you have the cetidine triphosphate, this end product here, cetidine triphosphate, and then uridine triphosphate, also inhibiting this second enzyme, aspartate transcarbamylase, this one. So you have these two, cetidine triphosphate and then uridine triphosphate, acting as inhibitors to this second enzyme, whilst you have the energy form, the molecular energy form, ATP, activating this enzyme. Then we also have the orotidine monophosphate decarboxylase, this enzyme, OMP decarboxylase, which can also be inhibited by uridine monophosphate. So you see this reaction from the OMP to the uridine monophosphate. This product, the uridine monophosphate, also has a feedback mechanism inhibition on OMP decarboxylase. Similarly, you have cetidine monophosphate. So either you have the uridine monophosphate or cetidine monophosphate, which will be acting as the inhibitors to OMP decarboxylase. All of this is what? Feedback mechanism. So you see this kind of feedback mechanism. Whilst you are reading biochemistry, you see such things happening over and over and over again. Okay, now, the degradation of pyrimidine. So it's just similar to what we saw with the degradation of purines. So you see, first things, dephosphorylation, that is phosphate group is removed from the sugar. And then there will be what? Deamination. The amine group is removed from that nitrogenous base. And then what? Cleavage of glycosidic bond. The breakage of the bond on carbon 1 of the sugar and the nitrogenous base. So it's just a matter of breaking these components of the nucleotide apart. Remove phosphorylation, that is this phosphorylation, amine group on the nitrogenous base. You remove deamination, and then the cleavage of the glycosidic bond between the base and the 5-carbon sugar, that the glycosidic bond is broken as a form of degradation. Then the bases will go degraded further. So you see those bases are having some amine groups on them, and you also appreciate the fact that you have amino acids coming together to form this ring. And so this degradation will go back into the formation of an amino acid, beta-alanine, and then some beta-amino isobutyrate, which is a derivative of an amino acid. So it's an amine group, amino, then some 4 carbons. So isobutyrate, it's like a carboxylic acid group. So isobutyric acid, which is also being referred to as isobutyrate. Then, as those groups are removed, then of course the amino acid, we saw the metabolism of amino acids, and so that amino acid will then undergo some transamination and deamination. So the amino groups will now be removed, transferred. Breakdown of this beta-alanine, it's an amino acid. Amino isobutyrate is also an amino acid derivative. And then once you have deamination, transamination, it means you are left with a carbon skeleton. And that carbon skeleton then goes into the formation of what? Acetyl-CoA, which will then go into the TCA cycle, or succinyl-CoA, which will also go into the TCA cycle to generate energy. Or you have a salvage pathway, just as we saw with purines, where the pyrimidines are recycled as precursors so that they will now form their respective nucleotides just by binding onto a phospholipase. And then once they bind to this phospholipase, they becomes what? A pyrimidine as the salvage pathway. Now, we talked about some disorders when we're talking about the breakdown of purines. And we mentioned the fact that the accumulation of uric acid can go into the formation of the salts of uric acid, which can get deposited in the joints, causing the condition of gout, or the elevation of this uric acid itself in serum, causing a condition of hyperuricemia. Now, in the case of the breakdown of pyrimidines, you saw in that reaction part, the intermediate compound or orotate, and I said this is also referred to as orotic acid. Now, the elevation of the concentration of this compound, orotic acid. So take note, in the breakdown of purines, we talk about elevation in the amount of uric acid, but here we are talking about what? Orotic acid. And the elevation of this orotic acid in urine can cause some retarded growth, some severe anemia. So what it means is that the like a similar effect that you are seeing with uric acid increase in blood. If you have this time around, it is orotic acid, and this orotic acid is having a similar effect like on the nervous system as we saw with uric acid, and to have effect on the production of red blood cells or some hemolysis of red blood cells. And so you now be having some severe anemia, increase of this orotic acid in urine. And of course, if it's having effect on the nervous system, then it can also cause some mental retardation or retardation in growth in general. And what will be causing such a thing? So you see the deficiency of orotate phosphoribosyltransferase transferase, this enzyme. So this reaction path starts all the way to orotic acid and continues from here. And the conversion of orotic acid to the orotidine monophosphate, that is the conjugation. So here you see that here you are having only the orotic acid, but here you see ribose 5-phosphate conjugated on N3 to start forming the nucleotide. And you see the enzyme which is responsible for that conjugation of orotic acid to the ribose 5-phosphate here, this orotate phosphoribosyltransferase. This enzyme, its deficiency will mean that there will be increased levels of orotic acid, because it will mean orotic acid will not be able to be converted to the orotidine monophosphate to go down the reaction path, and so it will start accumulating in the system. And similarly, the OMPD carboxylase, this enzyme, OMPD carboxylase, which is also responsible for the conversion of this orotidine monophosphate down the path to uridine monophosphate to then go down the whole path. So any deficiency in these two enzymes can be causing the buildup of orotic acid in the system, which will be referred to as orotic aciduria. In the case of purines, we saw hyperuricemia because it was uric acid which was accumulating. Here we are having orotic acid accumulating. Then there can also be, so this is like what we saw with uric acid, or even as, together with orotic acid, similar thing, being referred to as the Rez syndrome. But this one is referred as a secondary orotic aciduria because it is not being caused directly by these enzymes which are involved directly in the breakdown of orotic acid in the formation of the deoxy and the oxy, the deoxyribonucleotides, like here we are talking specifically about the enzymes which are involved in the conversion of orotic acid, the orotate phosphoribosyltransferase, and then the OMPD carboxylase when we were talking about orotic aciduria. But the secondary orotic aciduria, which is referred to as Rez syndrome, is caused by a defect in another enzyme, ornithine transcarbamylase, which is found in the urea cycle. And the urea cycle is beyond the scope of our course, so we will not go there. And because of that, of course, if you are going to be having, the conversion of orotic acid down the line, and this enzyme ornithine transcarbamylase is found in that urea cycle, which is having some effect, then of course it is going to cause the build-up of orotic acid. So you have an accumulation of carbamyl phosphates, which would then go into the direction of increased synthesis, and then of course excretion this uric acid to increase in the blood serum and as well as be secreted in elevated amounts in urine. And that will be the disorder referred to as the Rett syndrome, also known as the secondary uric aciduria. So that is the end of our lesson on the metabolism of nucleotides or nucleic acids. So I'm allowing you now to be able to unmute yourself so that if you have any contributions, any comments, any questions, you can raise them, and then we discuss quickly, and then we can end today's. We've done well. We started from 7, it's now 8:26. Usually what happens online is when you start lectures like this and it's getting too long, people start falling out for various reasons. So it's good that we'll be able to spend about most 1 hour, 30, 40 minutes, then that's okay. All right, the floor is open, so I'm listening. If you have anything to say, I'm listening. Everybody's listening. If you have anything to say, you can raise your hand, I'll call your name, and then you unmute yourself, you speak. When you finish speaking, then you mute yourself back. No hands, no questions, no comments, no contributions. If you have anything to share with us. All right, so if there are no questions, there are no comments, there are no contributions, because maybe when I was teaching, some of you came in later and did not hear what I said earlier on, I will repeat it, that I have put some questions under enzymes on LMS. And so you can go there and try your hands. And over the period this week, all the way over the Easter holidays, I'll be putting more questions under each of the sections. So carbohydrate metabolism, lipid metabolism, protein metabolism, and the last thing which we have done now, the nucleotide metabolism. I'll be putting questions under each of these sections. And so please try your hands on these things as you prepare for your exams, which will be coming right after you come back from your Easter holidays. The exam is going to be 60 objective questions, 1 for each mark. And it is going to cover all the sections from enzymes to the last thing which we have done now. You have questions under each of these sections for you to answer. Is that clear? So please, those who have not yet paid their fees in full and they don't have access to LMS, I will urge you that you please do so and have access to all the materials which are there, because it will greatly help you in your preparation towards the exam. Okay, so if there are no questions, again, if there are no, okay, let me see. I think there's one, I'm seeing someone there. Is it a chat? Check and see if someone has written something. Okay, somebody, okay, Albert says, can we get this slide? Of course, you get a slide. I will share the slide and I will share the video as well. We are recording this lecture. So I will share it after this, and I will also share the slide with you with some literature in addition. These things are, I think, the slide and the literature already are uploaded on Elements, but I will share it again with your rep, and your rep will share it with you. All right. Any other? Okay, so if there is other,
sir.
yes. Angela.
Yes, sir, please. I think there's also an issue with the course registration on LMS. So for me, I have registered, but I can't find the biochemistry theory as part of the courses here.
Okay, yeah. If you see such a thing happening, I will advise you to go to the IT department. Okay, I have seen that happening with some students from even other departments.
Okay, yes, sir.
So you go there, and then they will have a problem for you. Yeah, Sarah, you want to say something? Sarah Nita.
No, please. We thank you for your time and all that you've shared for us.
Okay. Thank you too for having the time to attend lectures from beginning to the end. So if there are, so I want to urge you that none of you should be afraid of exams. Everything which I have given you is just enough for you to prepare for exams. So just study with the exams, go on LMS, try the questions over there, have a better understanding of them, and the exams will be okay. Everything will be fine.
Yes, sir. Thank you, sir.
All right. So, any other? If there is no other, may I move the motion for the adjournment of this meeting? Nobody is seconding.
Yes, sir. I second.
All right. So we call it an evening, and have a good night.
Same to you.
Bye.